Hello and welcome to Neuro Navigator. Today's topic is visual processing. Same as with the Action Potential series, this topic has multiple parts. In the last videos, we've talked about eyeball anatomy, photoreceptor transduction, and then we talked about where the photoreceptor senses information. In this video, we're going to talk about what the brain areas do with that information. We previously talked about how light can trigger changes in photoreceptors, leading to activation of various cells later on. But after these ganglion cells are activated, where does that information go? Well, their primary target is the optic nerve. The optic nerve travels outside of the eye, and then part of the retinal tract will cross over at the optic chiasm, the nasal retina closest to the nose. The temporal retina cells, which are on this side of the eye, will stay on the same side. This means that the optic tract is carrying the axons of cells that originate in both eyes, so that the images in your left visual field are projected through the right optic tract and vice versa. Retinal ganglion cells will first synapse in the lateral geniculate nucleus in the thalamus, which you might remember is the relay station for much sensory information. The LGN is laminated. It's split into magnocellular layers, the parvocellular layers, and the coniocellular layers. These layers are made distinct by the retinal ganglion cells that are sending input to them. Ganglion cells are the largest with the larger receptive field and don't transmit color. K ganglion cells are the next largest and may deal with some color vision from short cones. And P ganglion cells are the smallest and convey color information. You might recognize there that we have the first letter of each of the cellular layers. After it synapses, that information travels through the optic radiation, still represented the same now as it was arranged in the retina. After that, it does get worse. The primary visual cortex is also layered, with the organization separating populations of neurons with distinct connection patterns. Now that can feel very conceptual, but largely what this means is that the many inputs are converging onto fewer cells as the regions get more specific in what they do. One cortical cell responds to the excitatory inputs from several LGN cells, which is how cells can end up being selective for dots or bars of light. Overlapping receptive fields can result in selectivity. Simple motion detection starts in the primary visual cortex, and then details can build from there. Area MT neurons can process the component signals of V1 simple cells into a more accurate perception of an object's movement. These cells have preferred directions and require some correlation between the cells to accurately identify motion. Neurons can also be selective for things like color, shape, and texture. In the inferior temporal lobe, neurons respond to facial features, and area V4 is responsible for color vision. This is far from a comprehensive mapping of the visual system. Other brain areas process other facets of your surrounding, even for things like your circadian rhythm in the suprachiasmatic nucleus, your pupillary light reflex in the olivary pretectal nucleus, and your ability to coordinate eye and head movements to a visual target with the superior colliculus. All of those brain areas are being fed information as well, but the most basic visual pathway goes photoreceptor, ganglion cell, LGN, B1. If you want to know more about those other structures and pathways, you're going to have to beg me.